Welcome to Scientist Stories. My name is Jyoti Sahi, and today we're speaking with Dr. Alok Khorana, Chair in Oncology Research and Professor of Medicine at the Cleveland Clinic in Ohio. Dr. Khorana is also the great-nephew of Dr. Har Gobind Khorana, who won the Nobel Prize in Chemistry, and January 2022 happens to be the birth centennial of Dr. Har Gobind Khorana as well. Welcome. Thank you so much for taking the time to speak with us today.
Thank you for the invitation. Yep.
Absolutely, absolutely. I just wanted to start by asking you what led you to your career in science and then subsequently to the field of oncology.
Yeah, that's a great question. So I grew up in India, in Gujarat, and both my parents were faculty members in the university that I went to medical school in, MS University. Mm-hmm. My father was a professor of psychiatry, and my mother was a professor of psychology. So when I was growing up and my parents asked, how was your day? It was very different than, other parents. And so there's always this push, as there is in many Indian and Indian American families, to sort of, work towards science. And medicine seemed like a natural outlet, just given my own family. We actually lived in a sort of subsidized housing for faculty members called doctor's quarters, which is right across from the medical school. So almost everybody I knew was a child of a physician or had at least one parent, if not two parents, that were physicians. So it was an unusual upbringing, but I think it definitely influenced my career choices. Oncology was a more sort of unconventional choice. So I knew I wanted to go into medicine. I knew I didn't want to be a surgeon because I just didn't have the sort of the physical ability to be a surgeon or skills. And so I wanted some type of a medical specialty. And I grew up my whole life with my parents saying, oh, everything's, psychologically related or is psychosomatic. And so my rebellion was to say, I'm going to choose the one field, cancer, which is not psychologically, related. And of course, that's wrong too, because cancer patients have so much mental stress and emotional stress. And I actually work closely now with this field of psycho-oncology here at our cancer center. But that was my sort of adolescent rebellion, was to sort of go into oncology rather than psychiatry or psychology.
Yeah, that was your little area of rebel.
Yep, exactly.
Very nice. And I know you're heavily involved in research, so can you just share with us what type of research you're involved in these days?
Yeah, so I actually work in two different aspects of oncology research. One, which I'm sort of better known for, is a very specific complication of cancer called cancer-associated thrombosis. And what that is that many cancer patients, when they're on treatment for cancer, either surgery or chemotherapy or immunotherapy, any of the newer agents, develop blood clots, either with the cancer diagnosis or while they're on treatment. And I found that really fascinating because nobody's actually investigated to the extent that they should why blood clots are so common in cancer patients. And so during my early training, I went into a lab and I studied sort of different aspects of coagulation and how it relates to cancer biology. And at that time, both our program and many other programs were sort of identifying that many of the molecules that are important for clotting, so for coagulation activation, are also important for cancer cells to be able to metastasize, to form new blood vessels, and to escape immune surveillance by the body. And so this sort of interdisciplinary connection between clotting and oncology was very fascinating to me. And so I've worked now for close to 20 years in this field. I've done a lot of epidemiologic work, identifying the incidence and prevalence. My colleagues and I developed a risk prediction tool, which is sort of now known colloquially as the Khorana score, that helps predict which cancer patients are likely to get blood clots. And then I participated in large clinical trials to both prevent and treat blood clots. And we're actually participating, leading one new anticoagulant in this field right now. So it's a very evolving field. To me, it's intellectually very interesting and obviously very meaningful for cancer patients because blood clots are actually the second leading cause of death in cancer patients after cancer itself. So that's one aspect of my work. And I would say that's definitely been major part of my research career. The second aspect is gastrointestinal malignancy. So I direct the GI cancer program here at the Cleveland Clinic. and most recently, I've done a lot of work with drug therapeutics for cancer patients and novel therapeutics. And most recently, we've become very interested in why younger people are more likely now to get gastrointestinal cancers, and specifically colorectal cancer. It's actually a growing problem both in the United States and worldwide. It used to be unusual for people less than 50 years of age to develop colorectal cancer. It's still, thankfully not super common, but the increase is much greater in this population. If you look at sort of, we see all these great headlines in the newspapers that cancer mortality is declining and cancer incidence is declining. And that's true if you look at sort of the whole population. But in colorectal cancer, if you divide younger than 50 and older than 50, we are seeing two opposite trends. People younger than 50 have an increasing incidence of colorectal cancer. People older than 50 have a decreasing incidence of colorectal cancer. So there's sort of an epidemic that's hidden by the improvement in mortality in older people. And so we're investigating, we formed a center for investigating young onset colorectal cancer and other GI malignancies. And we're trying to figure out, is it microbiomic differences? Is it proteomic differences? Are there environmental factors? Are there dietary factors? We're trying to sort of figure that aspect of it out so that we can help deal with this problem as it emerges and hopefully be able to correct it in the future.
Wow, that's fascinating. Very nice.
Thanks.
I wanted to just shift gears a little bit. You wrote an article earlier this year about your great uncle and Nobel Laureate, Dr. Har Gobind Khorana. It's a beautifully written article, and I just wanted to know if you could tell us a little bit about your great uncle.
Yeah, so he's, somebody that sort of loomed larger than life, for me my whole life. He won the Nobel in 1968 for deciphering the genetic code. And these days, it's sort of a given that we would know what the genetic code is because we've moved so, the molecular biology has advanced so far past that. But, going back to sort of the 1950s and 60s, DNA had just been discovered. The structure of the DNA, which is a well-known science story, right? The double helix and Watson and Crick, and sort of this whole notion that Rosalind Franklin, who was a well-known woman scientist, but was not credited for initially, at least, for deciphering the structure of the DNA. All of that happened in the early 1950s. And that was a big scientific advance, but it actually didn't lead to sort of true molecular biology because the chemistry of it wasn't understood. So it's great that they understood the structure, but how do you take this double helix and how does the body take the double helix and the information contained within the double helix and transform it into amino acids and then into building proteins? And that part was not known. And so what my uncle did was he came from a chemistry background and he applied sort of his chemistry knowledge to biological questions. So very interdisciplinary approach, and that was unusual at the time. And he worked with a lot of biologists. They were actually grateful for his work because he brought in sort of a novel perspective into understanding this. And he, along with other very key figures, Arthur Kornberg, who discovered DNA polymerase, Marshall Nirenberg, who also was crucial to deciphering the genetic code, they kind of figured out that, it's a triplet. So you need sort of 3 of the letters, 3 of the bases to translate. And then also that there were start codons and there were stop codons and how the messages were translated and so on. And so that work led to him getting the Nobel Prize in '68, and he was only 46 at the time. So obviously huge inspiration. It's a great science story, but I think beyond that, it's actually a great immigrant success story. And it's also, not widely understood now. But at the time in India, so remember, this is India just maybe 20 years after independence, at a time when the country was, had not fully realized sort of the promises of independence as many had expected that they would. And looking back now, I think we are dealing with a lot, post-colonialism that we hadn't quite grappled with. We had rid ourselves as Indians of the trappings of colonialism, but we hadn't quite grappled with what post-colonialism was. How do you come back to a culture of, that actually had a lot of science and had a lot of mathematics going back into sort of pre-modern times and medieval times, but all of that had been shut down for a period of 500 to 700 years. And so he was the first Nobel laureate for independent India. And this was a big win for Indians. And I think people perceived it as a big win. It was widely covered in the newspapers and newsreels. And, there was only like one television channel at the time, but on that one television channel. And when he came back in 74, when I was 2 years old, it was, I don't remember anything, but it was a sort of a hero's welcome and, covered widely in all of the newspapers. And he was sort of a celebrity at the time, and also very inspiring to other younger Indians and also parents of young Indians to say, oh, this is a path to success. Let's emphasize education. Let's get you the best training we can. And if we can't get you to succeed in India, let's just get out of India and into other countries where our potential can be fully realized. And that's a template that was not fully understood prior to that and became fully understood his career was used as a template for many other, I would say thousands of Indian Americans and Indians emigrating to other countries, Canada, Australia, the UK, and so on. So inspiring from both the science perspective and then also inspiring from just this sort of post-colonial, post-independence, let's return to our roots of success and triumph that's been established over millennia, but that we've lost for a few hundred years.
Can you share with us how, I find it fascinating that a child with no school in his hometown, you mentioned this in the article, ends up moving on later in life to win a Nobel Prize. That is just fascinating. Can you share a little bit about that?
Yeah, I actually distinctly remember because my own son was, I remember having this conversation with him because I was kind of stressing out. My son was like 2 years old at the time, and I was trying to decide, should I put him in like a Montessori class or, like a regular, type of kindergarten. And I was just sort of chatting with him, and he disclosed, he's like, well, I didn't go to school till 8th grade. So to me, that was, it was actually really clarifying because I was like, we stress out so much about these small decisions, but, it's not really the type of schooling. It's just getting people to a place where, everybody has a lot of potential. And how do we get people to a place where that potential is recognized and where it's allowed to flower? Unfortunately, at the time, India was not in a position to help realize that potential. He came back, not only from, it was great that his talent was recognized, at least in high school. He was sent to university, and then the university selected him for a scholarship to go to the UK. So there was some form of talent recognition. But then he came back from the UK with all this great training in what would, subsequently become sort of the whole field of molecular biology. But he was sort of tied by this bond to the Indian government, and he was supposed to work in the Indian Agricultural Research Institute. And there were no jobs for him because his training was sort of beyond of what the infrastructure at the time allowed. And so he hung out for a few months, and then the government released him from his bond and said, okay, yeah, you can go back now because we don't really have a position for you. And then he ended back up in the UK. Perseverance, I think, was a big part. So, he didn't get an academic job straight off after his postdoc fellowship. He ended up in a non-academic job in Vancouver, which he didn't even know where that was. He had to find it on a map, but he accepted the job. Flew out to Vancouver, and he converted this non-academic job into, the work that led to the understanding of the genetic code started in that lab in Vancouver. And it's a joke that they kind of hired him because they figured an organic chemist would be less expensive than, some other type of biologist because all they needed was beakers and test tubes. Not any fancy equipment. And so there wasn't as much support as he would have liked, but he, again, it's all about taking the opportunities you have. I think immigrants are very good at, if we get access to the playing field, even though the playing field may not be level, we're very good at using whatever access we get into, into opportunities because we know how challenging some of these opportunities can be. And so, he's alone in Vancouver, no real sort of environment surrounding him with molecular biology. But he used the skills that he had picked up in the United Kingdom and in Switzerland. He had taught himself German, and he used, and at that time, a lot of the scientific literature was in German. And so the German scientific community had developed some of these enzymatic methods to synthesize, that could be used to synthesize certain compounds. But nobody in the English-speaking scientific community was using that. And so he used his German skills to say, hey, we could try this. And he tried it. and it worked. And so he synthesized ADP and ATP. I mean, these are such common terms now, and nobody knew how to synthesize it. And then he synthesized coenzyme A, and he realized he really had a lot of skills in this type of using chemistry approaches to biology, building biological molecules. And then, once he started publishing this, a lot of famous scientists from the US came and visited him. Arthur Kornberg came from Stanford and spent a couple months with him in Vancouver. And then that led to an invitation to move to Wisconsin. And then he got the Nobel in Wisconsin and, went on to many other things. So it's really, I think, just recognizing potential, and, a lot of people have potential. Unfortunately, in many environments, it's not recognized. And then allowing, that potential to flourish in an environment that sort of best works. So maybe just getting out of people's way, I think, is sort of.
But one thing I learned that he went to Switzerland on his own money. He has saved in UK because he was so determined to learn the German. And it was ITA, I think, and that's where, he learned the German, and whatever saving he has, for a year or two, I think, he used it, and then he moved to Vancouver.
Yeah, that's exactly right. So he showed up in this lab in Zurich, Vladimir Prilog, who actually went on to win the Nobel Prize himself a few years later. He had no actual job. He just kind of showed up and said, "Oh, I have these skills. Can I help out in your lab?" And they said, "Well, we don't have a stipend for you, but you're welcome to hang out." And so they gave him like a little small space. And actually, for some weeks, the story is that he was actually just sleeping in the lab because he didn't have a place to live. And again, it's about once you get into the playing field, you'll make the most of it that you can. But and some doors are open for him, but he opened a lot of doors on his own. And he's always credited that 1 year in Switzerland with understanding sort of true molecular biology and true real science, and being able to carry that forward in his own work in Vancouver and then in Wisconsin. And also, that led to him sort of finding the love of his life, his wife Esther, who was a Swiss-born person. They had met actually before when he was a postdoc in the UK. And then they got married in 1952, right before he moved to Vancouver. So, and his 3 children were all born in Canada.
Oh, very nice, very nice. What you like you mentioned, he won the Nobel Prize when he was just 46, and yet he still continued to pursue and achieve in science. And like yourself, you've been in oncology research for so many years. What do you feel keeps you, kept him motivated? What keeps you motivated when you are trying to figure out these, big problems in science or solutions?
Yeah. So I think it's finding work that's intellectually challenging and stimulating, and finding problems that haven't been solved yet. He has this epigraph in his, so he didn't write an actual autobiography, but he did sort of an annotated sort of best papers book, selected works in chemical biology, I think was the title. And the quote he has on the front page says, "We must be modest in everything but our aims." And that's really, if any one quote can capture what he did, I think that's really it. He was a very modest person, very humble, didn't brag too much or anything. It's a very different scientific personality than the Western world is used to. I want to point that out. I mean, this sort of team science approach, interdisciplinary approach, remaining humble, sort of staying rooted and grounded. These are not, look at sort of James Watson, for instance, very different personality. Crick, who was part of the DNA, the double helix thing, very different personality also. In fact, they were so confident they were going to crack the genetic code that they created something called the RNA tie club. And they all wore sort of ties that had like the RNA structure on it or something. And this is without even cracking the code. And then for the next 10 years, they couldn't actually figure it out until, my uncle and Nirenberg and Holley and Kornberg sort of put their heads together. So, I think part of it is just doing it sort of for the work and not for the accolades, just saying, this is really interesting, let me try and figure it out. And then the accolades may or may not follow, but really to me, the work is what's interesting. And you kind of see that in his career. So, a lot of people would rest on their laurels after winning the Nobel Prize, but he went on to work on synthesizing the artificial gene. That was the first time that was done. That was a huge deal. And that happened a few years after. And it's not as well known, but the concept of the PCR, the polymerase chain reaction, which, everybody who's had a COVID test, which is pretty much the whole world right now, that test is still widely used now. And that's not the only thing it's used for. It's used for, there's hundreds of, if not thousands of applications. But the concept of the PCR also came from his lab. There was actually a whole lawsuit between DuPont and Roche around who actually invented the PCR. But everybody agrees, and Arthur Kornberg in his autobiography is very clear about it. He was actually a witness for whichever company didn't have the patent to say that he believed that the concept of PCR came from the Khorana lab and not from one of these big corporations. So there's tons of yes.
I remember reading that paper. It's published in Journal of Molecular Biology with a couple of sentences that, you take the polymer, you do the annealing and then you do the polymerase, and then therefore you can make tons of DNA as you wish.
Right.
I was in Boston at the time, and I remember that very well. He deserved another Nobel for that alone.
Yeah, that was in 1971. And there was a Norwegian postdoc in his lab. He was the first author, Kleppe. Actually, he and his wife was also co-author on that paper. So there were two Kleppes on that paper. And then my uncle was the senior author. But you're absolutely right. They clearly laid out what PCR would be. So conceptually, they laid it out. The challenge was at the time, thermostable, Taq polymerase wasn't available. And so that's why they couldn't actually do it, because there was a heating process and a cooling process.
Yeah.
And so it was too cumbersome to do it until the thermostable polymerases came along, which took another decade and a half. But in terms of, who actually invented it, I think there's a lot of controversy around it. It's actually a great story. There's a whole book around who invented the PCR. The Nobel went to Kary Mullis, who worked for this corporation called, I think, Celtech. He got the Nobel for it. They were also upset because he was only one of the people involved in developing it within that corporation, and he actually also didn't do the sort of the true PCR the way eventually it ended up being doing. So they felt other people in their lab should have gotten credit for it. And then the biology community felt that not just the corporation employees, but also, the Khorana lab should have gotten, Kleppe and Khorana should have gotten credit for it as well. But that's not how it worked out. But, we can still look at the history and understand how this happened. So it's not just, the genetic code, but it was also the synthesis of tRNA and artificial gene synthesis, basically. And PCR. I mean, these are some really major accomplishments. And he was nominated for additional Nobel Prizes even after the first one. And then in late 70s, he switched his field completely and started working on G protein coupled receptors and photoreceptors in the retina, which had nothing to do with molecular genetics. And he worked on that field for another 20 years or so. So I think driven primarily, speaking for him, driven primarily by intellectual curiosity, I would say.
Yeah, fascinating. What advice do you think Dr. Gobind would like to give future generations, and what advice would you, from your experience, like to give future generations?
Yeah, that's a great question. I don't know that I can speak for him. I think he was very clear cut in in his feelings that this sort of again, I go back to sort of be modest in everything but your aims. So, don't sort of brag and, about things that you're accomplishing. Do the work quietly, do it well. It doesn't have to be in science, although I'm sure he would have wanted it to be in science. It could be, in politics or literature or art, whatever it is. But, really, make meaningful contributions to society and stay grounded in your sense of who you are. And work in teams. That science is a team sport. I think we recognize that now more than we did several decades ago. And I think science is the better for it. But the way he conducted his research, I think, is an inspiration in being interdisciplinary, sort of intertwining, chemistry with biology, working with pure biologists and organic chemists and all of these different disciplines. I think is just really a fascinating way to conduct science.
Yeah. And last but not least, I heard that you are quite an avid reader. So if you could recommend one book for the younger generation to read, what would you recommend?
Oh my gosh. Okay, I can't recommend just one book. So the book that's affected me a lot in the past several years is this book called Overstory, which is about trees. And it actually sort of brings in climate change and the place of humans in society. I think this is a very new concept that humans center themselves in every story and in every narrative. But we are just one species on a planet teeming with billions of species. And frankly, you could argue that, the planet would be better off without us as a species, based on sort of all the destruction we are causing and specifically with climate change. And what Overstory does is it actually centers trees as part of their narrative. In a way that I've not seen done in literature before. It's very cleverly done, but also very authentic. And it really made me rethink about sort of, how we construct civilizations and how we construct our cultures, and appreciating sort of non-human aspects of the planet. So if there's one book that I would recommend, I guess that's the one. So
wonderful. Well, that's all I have. It was an absolute privilege to speak with you today. Thank you for sharing your story, and thank you for sharing Dr. Gobind Khorana's stories.
Thank you so much, Maria.